Hello students, now we are going to discuss about the another mode of classification, that is called morphological classification, right? So when we talk about the morphological classification, as the name indicating that it is based on the morphology, right? So in this classification, the drugs are arranged according to the morphology, right? What is the morphology of that particular drug? That according to that, when we are going to arrange the drug, like it may be the external characters, right? So what is the external character of the plant part? Maybe root, stem, fruit, flower, or any animal parts. It may be like called liver oil, like or any hormone, any enzymes, trypsin, chemotrypsin, like that, right? So here example maybe leaves, roots, bark, and stem, right? So these are the drugs arranged according to their alphabet, so that it will be easy to remember. So you just go through, which you will definitely find the easiest way to remember, because some tricks has been also involved over there and arranged according to the remembering point of view, right? So when we talk about the example, remember examples are very important here, because when we give example, more number of examples, we are creating the unique answer among the other students. So that's what the getting of good marks, the chances will increase here, right? So that's what the first, it has been divided in two category: organized and unorganized crude drug, because it is morphology, or we are seeing the external feature or external characters of the particular drug. So first leaves, right? So these leaves, it is arranged in the alphabet. Once again, one from A there is no leaf, means it is there, but we are not studying that one. So in the alphabet B there is a belladonna, which is very popular, atropa, belladonna, atropine we are getting. So that's what this drug I am keeping first. Belladonna. C for coca leaf, very popular. Digitalis, it is very popular. Digitoxin and digixin, these are the chemical constants we are getting. Then eucalyptus leaf is also very important, right? Eucalyptol we are getting. Eucalyptus oil is very famous, right? Then senna leaf we are using in practical also. Senna, Calycia senna, right? Alexandrian senna. Then tulsi leaf, it is very popular, not even in the Pharmacognosy, it is very popular medicinal plant in India. Vinca, very famous, vincristin and vinclastin we are having and we are using, right? Coming to the bark part, here also it has been arranged in alphabet order, so that it will be easy to remember, like Arjuna bark, Ashoka bark, Cinchona bark. From A to from C to Arjuna, Ashoka, Cinchona, cinnamon, like that you can remember. And Cascara and Kurchi also you can mention if you are able to remember. Roots parts, ashwagandha and rauwolfia is very popular drug in B Pharm course. We are going to study like vithanolides are very important here. Reserpine, deserpine are very important chemical constants, antihypertensive drug, right? So these two drugs you can mention in the root. Rhizomes, what you can mention, ginger we are going to use in the what you call starch grains measurement, and in the lycopodium spore method, ginger powder we are using in practical. So ginger you can mention, you can correlate from the practical in rhizome. Turmeric is very popular, not only for pharmacognosy, just like a tulsi. Turmeric is also very popular Indian medicine, and it is having huge history also. Seeds, when we talk about, A for almond we are having, B for black mustard you can have, C for cardamom. In alphabet, if you try to remember what is there in the seeds, right? So it will be easy to you. Isabgula, it is, we have used it for the swelling index and swelling factor. Linseed is there. Nux vomica seed, very popular. Strychnine, brucine, it will be there in that, which is popular anti natural pesticide. Coming to the woods part, wood, kochia wood is there, sandalwood is there, right? In fruit we are having amla, bale, then cardamom fruit, dill fruit, fennel, coriander, tamarind, capsicum, behra, lots of fruit are there. But if you try to remember by heart them in alphabet, like A for amla, B for bale, C for cardamom, it will be easy for you to remember in exam. Flowers and its parts, when we talk about pyrethrum flower, very famous. Saffron flower, clove bud, it is also flower, right? So flower part actually. So in fiber we are having cotton, jute, hemp, flax, right? Then when we talk about unorganized, this in this morphological classification we can write the huge number of examples, like dried latex, we are having opium, we are getting the opium, morphine, codeine. And from papain we are having carica papaya, from we are getting papain enzyme. Dried juice we are having aloe and quinoa, right? Then dried extract, we can write the black catechu and pale catechu. Agar can be useful. Gum, we are having acacia triga, again it is there in the unit 4, so this you can write down. Even agar is also there. Resin, asafoetida, that is hingalu or hing, that is there. Benzoin, like dhupam, nothing but, okay. So A, B, asafoetida, benzoin. Volatile oil, coriander, A, B, nothing is there, coriander. Then eucalyptus, very famous, we have seen into the here also in leaf example, here also in oil also you can write down. Fixed oil, chalmongra oil, coconut oil, linseed oil, groundnut oil, sunflower oil. There are huge list of the morphological classification, right, in example. Now the next mode of classification we are having, that is nothing but chemical classification, right? So as the name indicate, chemical classification. In this classification, crude drugs are classified according to their active constituents such as alkaloid, glycoside, tannin, etc., right? So what kind of chemical constituent it is there in the main quantity or huge quantity, and the drug is famous because of the presence of that chemical, and it's having its own therapeutic value because of the presence of that chemical constituent. So according to that, when we are classifying, it comes under the chemical classification. Let's try to understand about the examples. Examples are very, very important in when you are writing the classification of crude drugs. When we talk about alkaloids containing crude drugs, we are having A and B, we don't have. C for cinchona, D for datura, N for nux vomica, then Rolfia and the vinca. So these are the popular drug which are containing the alkaloid. Coming to the glycosides, in glycosides we are having aloe vera, we are having digitalis, ginseng and the senna. Coming to the tannins, we are having catechu and we are having myrobalan fruit. In resin section, we can write asafoetida, A for asafoetida, B for benzoin, B for balsam piru, balsam tolu. Side piru and tolu, these two you can remember. Balsam in there in the both the place. In carbohydrates, we can have acacia triacan. They both are brother, I told you in the class also. So these two drugs can have they'll go together, and most of the similarity it is there. Both are belonging from Leguminosae family, so that's what they will be together. Starch and isabgol also you can write down. Starch, we are having 4 types of starch, like we can say rice, maize, potato and this thing wheat, right? Then we are having isabgol seed, which we have used for swelling index in practical. So there, from there you can correlate that it is having carbohydrates. In chemical classification you can write down in the carbohydrate section. Coming to the volatile oils, we are having clove, cinnamon, right, coriander. So triple C BGF, right, like that you can make the formula. Clove, cinnamon, coriander, black pepper, ginseng, fennel, right? In lipid, we are having beeswax, cocoa butter, castor oil, cod liver oil, olive oil, coconut oil, sunflower oil, chalmongra oil. All those oils you can write down in this section. Enzymes, we are having papain enzyme, pepsine, trypsin, casein, right? So these are the things we have seen in the unorganized crude drugs also, right? So once if you will start by hearting these chemical constants, it will be easy for you to remember. Okay, now next mode of classification, it is chemotaxonomical classification. Very important, sometime it will be asked into the short notes, right? So write a note on the chemotaxonomy like that. Okay, so when we talk about the chemotaxonomy, as the name indicates, right, this system of classification relies on the chemical similarity of taxon, right? So chemotaxonomy, chemical similarity on the different taxon, right? It is based on the existence of relationship between constituents in various plant, right? So for example, this is the plant A and this is the plant B. So in this plant B, for example, these chemical constituents also the same is I have shown into the square shape, and this is also square shape. So what is the chemical relationship and what is the chemical variation? Like suppose this triangle, it is not there, here it is the circle, but this this square it is there, and in this plant also it is there. In plant A also square is there, in plant B also square is there. So there is a relationship, whereas the triangle it is there in plant A, and here circle is there in the plant B. So this is the chemical variation. So we are seeing the taxonomy also, at the same time we are seeing the chemical variation also, right? So let's try to understand with the example. When we talk about the example, propane alkaloid, right? So propane alkaloid, it will be there in the as a chemotaxonomic marker in Solanaceae family. All the Solanaceae family plant, they are going to biosynthesize the propane alkaloid. In the same way, rutin, it will be there in Rutaceae family. So this is common, but this routine will become chemotaxonomical marker, right? So why we do that? Because we need to know when we are going for chemotaxonomical classification, or we are doing chemotaxonomic classification, what are the possible action? What are the possible action between these two plants? Then biosynthetic pathway, what kind of biosynthetic pathway they are adopting among them. Then chemical variation, we are going to have this. So biosynthetic pathway also it is going to have what kind of biosynthetic pathway they are adopting. Chemical variation, right? What kind of, that's what I told you, triangle and circle. So what is the chemical variation between these two plants? And then chemical similarity, that's what this kind of similarity if it is there. So these things we need to know when we are going to classify the drug according to the chemotaxonomic background. Now next one is the next one and the last one, the seventh one, serotaxonomical classification, right? So when we talk about the serotaxonomical classification, this classification is for very similar plants by means of differences in the protein they contain, right? So here the protein is very important. Their chemical was important in chemotaxonomy. In serotaxonomy, sera means protein. So protein they contain. What is the difference in the proteins? We are not bothering about other things, root, stem, fruit, flower, or chemical constants or morphology or text. No. Serotaxonomy, it is very here protein we are important, our antigen it is important. The technique is based on the highly specific relationship between antigen and antibodies produced in the response to them, right? What is the antigen and antibody it is going to produce, right, into the plant? Proteins are carriers of the taxonomical information, yes, and commonly used as an antigen in serotaxonomy, right? So in plant protein, they are the carrier, means the transporter of the taxonomical information, right? So what that's what the arrangement of the leaf, the arrangement of the fruits, the style of the fruit, and biosynthetic. There are so many factors which is there. So protein is the basic unit which will give the information about the particular plant. It developed first in the Germany and became popular there only. Then later on all other countries they started using this particular system of classification. So let's try to understand with the one experiment which will be done actually into this system of classification. Experimentation of antigen antibodies, right? So mostly done with the plant. So what we are doing here is, first of all, one plant will be taken, and the protein from this particular plant, that is the parent plant, protein extracted from a plant, right? That is antigen. The protein it is extracted from the plant and injected into the blood of an animal. Let us take the example rat, right? So this will be injected into the one of the animal or rat we can take, where it become as an antigen, right? Here it will become as a antigen here, and with the response of this antigen, antibody will be producing. So after some interval, the production of antibodies it is going to taken place into the animal body, and then later on blood sample will be collected and compare the first plant protein or this antigen with the extract taken from the other plant, right? From here, because these two plant we are going to keep into the serotaxonomical category, so we have to check it. So here it is going to be checked in the particular laboratory, what is the response of this antibody or this antibody produced and this antigen, right? Because this antigen antibody already have the similar reaction. So according to that protein, based on the sera, classify the plant. So what is, suppose this antigen or this sera or this antigen, if they are looking to be the similar, so these two plant can be put into the one category as per the serotaxonomical concept, right? So this is also very important, and it has been added newly into the syllabus, and it will definitely sometime it will be asked separately, like describe or explain about the serotaxonomical classification, or what is the basic difference between chemotaxonomical and serotaxonomical classification. So definitely you should know that chemotaxonomical it is based on the chemical and taxonomy, and serotaxonomy it is based on the protein and taxonomy, right? So chemical constants and protein content both are the main thing here, right? And the remaining other classification, you know, morphology means what part, chemical classification in what chemical, pharmacology means what activity, taxonomy means what is the taxonomical background, alphabetical means name first letter, Like that we can have the basic thing. Thank you all.